All right, good morning, everyone. Welcome to High Rounds today on October 13th, 2023. We're lucky to have two speakers today. He's going to talk about the last gift, where we are now. So first of all, I'll introduce Dr. Robert Deiss. He's a physician at the Owen Clinic and an assistant professor in the Division of Infectious Diseases and Global Public Health. Prior to joining the Owen Clinic family in 2020, he was a research physician and site PI for the infectious disease clinical research program at the Naval Medical Center here in San Diego. He also spent a little over a year working in Mozambique with Doctors Without Borders just before coming to UCSD. He's earned some renown for his musical playlists, both during and outside of high rounds, and there will be a playlist today. Currently, he's co-director of the Clinical Outreach Pathology and Ethics, which makes a perfect acronym called COPE for the last gift study, and he will be sharing a bit about the study cohort and recruitment efforts to date. Our second speaker will be Dr. Sara Gianella Weibel. She's a associate professor at UCSD, specializing in HIV and translational virology. She has held positions of leadership within the UCSD CFAR as director of the Translational Virology Core, as well as the HIV Neurobehavioral Research Center as the co-director of the Neurovirology and Biology Core. And most recently, Sara won a 2023 Health Sciences Faculty Excellence in Mentoring Award, and we are pleased that she is working with all of us. And welcome to both of you. And I think, Robert, you're going to take it away.
Okay. Well, yes. Thank you, Dan. I can't imagine who put you up to commenting on my playlists. But yeah, I guess they're renowned, as you said. Yeah, and Sara, thank you again for joining us. We're very excited. Well, not joining, but being a part of this. And I'm very glad you're going to present some of our findings from the study a little later as well. So just to sort of go over the talk and how this will be structured. I'll just give a brief overview and a little bit of the background and rationale for the last gift study. I'll share some of the demographics of our participants and who we've sort of been recruiting and how that goes. I'll talk about some of the lessons learned that we have sort of accrued during the recruitment process and show some interesting numbers from that. I'll touch on the social and behavioral component of the study that many people aren't necessarily familiar with. And then Sara will sort of share some of our exciting findings that we've been able to put together. But I always, this is in honor of CJ Toby, the recent speaker we had during the Low Barrier series, who always started with gratitude. So I put together a playlist of gratitude. And Marvin, you might as well put that in the chat box now. Here's the songs on the playlist. So Kind and Generous by Natalie Merchant. Thank You by Led Zeppelin. Thank You by Dido. Thank You by Alanis Morissette. Thank You by Estelle. Thank You for Letting Me Be Myself Again. That's Sly and the Family Stone. Thank You, Busta Rhymes, etc. I want to thank you. And then Thank You Next. And of course, thank you for the music. But I figured this sort of list goes in concert with, the last gift study, right? And the gratitude that we have, for our participants, for their loved ones, in terms of, their altruism and their contribution to the study and to our science. Now, the playlist, as I said, is in the chat box. I don't know if anyone's familiar with the Wizard of Oz, Dark Side of the Moon, where if you put it together, so it's 44 minutes long. You're welcome to play it in the background, as this talk proceeds, and that's the link. Okay, so moving on, the rationale for the last gift study is that we've been looking. It's a cure research study, and to really get at that, we need to have a full characterization, understanding, and ability to access the reservoir for HIV virus. But this is not easy. The virus is established during primary infection in peripheral blood cells and in anatomic sites in solid tissues, all throughout the body. HIV, the virus, though, plays hide and seek with the immune system and ART therapies. And this is why when we stop medication, as everyone knows, there's usually a period of virologic rebound. But accessing these reservoirs is not easy. We conduct most of our studies in living people, and it's hard to reach the anatomic sites safely, such as CNS, such as the brain. So we could do autopsy studies, but those are often performed, hours to days later after a person will die, and that sort of changes the nature of the specimens, of the tissue, and it's going to be very difficult to collect live virus under those circumstances. The second sort of limitation of autopsy studies is that there's often sort of not really great pre-ante-mortem characterization. Data collected can be very remote before death, sometimes months to years. Excuse me. And for a cure research study, something more recent and is helpful. And so this was the last gift study was sort of the proposed solution to this issue, in that we sort of reached out. Davey tells an interesting story, actually, of one of his patients who helped inspire the idea, who had done research all his life throughout, throughout his adult life, been part of HIV research, et cetera, and then found that he wasn't eligible for studies, after, he received a certain diagnosis. And, it was almost a little bit offensive to the person. And so, we realized that there is a community of people who still want to, participate, contribute to research, et cetera. So that sort of has become the population for this study. Altruistic persons living with HIV who have a terminal illness, such as cancer, an AIDS-defining illness, or some sort of end-stage organ disease. So what we're able to do now, sort of by enrolling, this potential group of individuals, is to follow people for months, weeks, days, et cetera, whatever we can do to collect some of their clinical data, urine samples, vaginal cells, blood, et cetera. And then, the heart of the study, but it's not the only piece of the study, but that is to perform a rapid autopsy wherein we can collect organ, tissue, and fluid samples. There are examples of this sort of research, or this type of study in cancer research. And so the goal of the rapid autopsy is that our team performs sort of the sample collection, et cetera, the autopsy within 6 hours of death. This gives us the opportunity to collect fresh samples, capture live virus, et cetera. It preserves the quality of the RNA and proteins, that are later used for research. And so this is what an autopsy, will look like, right? This was photos taken from one in 2018. It's a very impressive sort of process and ordeal. Often, just by nature of the way, sort of healthcare works, these sometimes happen in the middle of the night, right? It's unpredictable. But the team, sort of is on call 24/7. We do often know that maybe someone's death is imminent, but not always. And even if we know someone is sort of approaching death, it could be at any point, say within the week. So generally we can prepare, but sometimes the time is sort of it's a mobilize everybody all at once. And Sara remembers once where it was my first day on the study and I got a call at 3 a.m. And really couldn't believe what I was hearing. So fortunately, I didn't need to go, and we had a good team that day. But so it's about 10 to 12 people who mobilize, quickly. And as you see here, sort of this is just what it looks like on the outside. We have one sort of the diener who, kind of prepares all the tissues, kind of does the actual cutting and the autopsy. And then our team of cutters here who sort of dissects the tissue even further. The diener, the sort of one who's cutting most of the body, will sort of bring over specimens and say, okay, this is the spleen, and then we dissect that further. One person, and when I was in an autopsy, this was my role, just sit and hold and collect tubes from the specimens that are being cut. And this is an impressive ordeal as well. There's between 800 and 1,000 different tubes, because multiple samples, say from each organ site, in order to be stored, preserved, sent to collaborators, etc. And then so that's a lot of the fresh tubes, a lot of the fresh tissue will go into those sort of tubes, and some will be stored fresh, and then some will be stored frozen, and there's sort of a process for that as well. And by all means, I don't know, Sara, if you have anything to jump in and contribute. You've been doing a lot of these, sounds like. No. By all means, if I like my talks to be interactive, so if people want to put questions in the chat or whatever.
But maybe the one thing that I can say is that there is only 20 rapid autopsy programs in the US, and Last Gift is by far the most extensive, because almost everyone else is doing it for cancer, and so they limit the tissue collection to cancer and surrounding lymph nodes, while the Last Gift collects 50 different anatomical sites. So it is really huge. The logistics behind it is incredible. It's very hard to replicate. It was a lot of work to put it together. And every single autopsy is unique. Like, you learn something from every autopsy. There is not two that are the same. And so really with experience, we learned, we are getting more efficient, and now we are a well-oiled machine, and I'm very proud of the team. It's incredible.
Yeah. No, thank you. And so that sort of leads into the next slide, that that it's not only the autopsy that happens sort of immediately, and the pagers go off, etc., and everyone goes to the Hillcrest sort of basement to do the autopsy. But there's a period afterwards that equally is as important, right? So sort of after an autopsy, and this is on the same day, our staff might get to decompress a little bit, sort of reflect on the experience, eat, right? Because it's been hours, since that's been happening, maybe. And then the team goes to the lab. Again, sometimes this is middle of the night. The specimens are organized and frozen, and there's a lot of cleaning that needs to happen. Interestingly enough, remember that this is collecting fresh tissue, and so some of those specimens will need to go out to the collaborators. They're on the receiving end, also, ready to prepare. They're having, they're aware in the middle of the night, or whenever. I mean, it's not always the middle of the night, but they're aware that, there's the autopsy is happening, and they need to be ready to receive. So our specimens go to Los Angeles, then they're shipped, to the receiving labs. And that all happens, very quickly. Now, throughout the week, then, obviously not everything is done right away, but, additional samples that are frozen can be shipped. And, the big one here is preparing for the next one. I mean, that sounds like, okay, well, we'll just get things ready. But honestly, it takes, over a week, if not a little more, to get all the tubes. I mentioned 800, 1,000 tubes. They all got to be labeled, ahead of time, and everything done that needs to, sort of help this process go smoothly. So, Yeah. Anyway, so it's a lot. But let me tell you a little bit about, you know, the study participants that we have recruited so far. So we have had 42 total participants in the study, 12 of whom are alive. And obviously, you know, this is only some of the, you know, information that we would collect, but I, you know, just sort of want to show you, you know, some of the important things that we look at, obviously, time on the study, et cetera. So the age of our deceased study participants has been, you know, our median age is around 62, but there have been young people, and, you know, all ages throughout. As far as sex assigned at birth, the majority have been male, and, you know, race and ethnicity, there's a sort of white predominance, you know, and so this is something we're looking at. I'll show you our current participants. We're making some efforts, you know, we'd love for our study population, obviously, to reflect, you know, sort of the you know, picture of people living with HIV in you know, in the US and the world, obviously. So, you know, it's a matter of outreach and you know, increasing our recruitment efforts for that. Among our participants, you know, who have already died or had an autopsy, their median time on the study was 3 months. You know, sometimes it's very short, sort of an in-hospital or inpatient enrollment, you know, on someone who's expressed this wish in the past. But as we see, you know, people can live very long and be on the study. And, you know, so there's sometimes a misconception about last gift that it's really for like someone on hospice who's dying right now, you know, and actually, as I mentioned before, we like to be, you know, sort of talking to them and, you know, collecting specimens along the way. So, you know, months, you know, is great, you know, and occasionally people live longer than we might have anticipated. There you see the causes of death, which tend to be cancer, end-stage organ disease, again, because those have predictable sort of disease courses. And, you know, and so naturally, you know, sort of lend themselves to a study like this. Oddly enough, we've had 3 patients with 3 participants with ALS, which, you know, seems, you know, just a little bit unusual. But again, it's sort of a disease with kind of a predictable decline, and you know, and it brings up interesting opportunities for collaborations with other sort of ALS research, and that's something we're excited about. 4 of the participants had some sort of AIDS-related, you know, diagnosis at the time of death. So, of our current participants, we have 12. Just as you see here, many of them are Owen patients, and we'll come back to that. But you know, sort of many different diagnoses, and here, very similar. The population or age range, you know, between 50 and 94, median age in the 60s again. Sex assigned at birth, where, you know, we've have a few more women that we've been able to recruit recently. And there's diversifying just a little bit with 3 African-American patients, 1 Hispanic patient, who sort of remain alive. And as you see, you know, the time on the study, we've done some recent enrollments, so that those are the people who are, you know, just on the study. We've been following some participants for 5 years. You know, occasionally we say, you know, this is a study that we have, and, you know, someone might turn the corner, get better. And I think it's an interesting question of whether the study motivates people sometimes to, you know, sort of either, you know, be better about taking medication, et cetera, and maybe even, you know, sort of live longer, outlive the study. That's something I tell people, you know, that they can always do. They don't need to, you know, this isn't necessarily a study where you're dying right now. And again, similar sort of characteristics as the prior group. So let me just talk about a little bit about study recruitment. And I put this sort of text box at the beginning here. This is a video that's on our website, which, you know, you'll have at the end. But you know, it's a very nice video, and so it's become one of our sort of recruitment tools, you know, in terms of our outreach. I've been alluding to this. So, who are the people who participate in our study? They will be people who are living with HIV and have a life-shortening illness. I took that language straight from the website. People who reside in San Diego County, and that's because of the logistics of getting a body to UCSD Hillcrest within a certain number of hours for us to perform the autopsy. There is an asterisk there because we are increasingly talking about sort of broadening the area and maybe trying to get Palm Springs involved or the desert. It's not easy, and we're sort of actively thinking about that. But again, so even though we say San Diego County, I add that qualifier. And I just want to say that, again, life-shortening illness can mean many different things. It doesn't have to mean days or weeks, as I sort of alluded to earlier. I would say, though, that the ideal scenario for one of our study candidates is someone who's accepted their prognosis and made or sort of began to consider or make plans for the end of life. So anyone who's certainly referred to hospice has had something of this conversation and makes participating in research something that they can consider. So some of the ways we recruit, there's community outreach. I mentioned the website. We have a brochure for it. We offer presentations to providers in the community. And then, of course, I'm part of the study team and work at the Owen Clinic. So there's some, word of mouth or just, some patients that I sometimes wind up seeing myself. We also take referrals from our parent study, which is the California Neuroaids Tissue Network, where people have sort of enrolled in a study, long before anticipated death, agreeing to donate brain to research. And then some of those patients become sort of candidates for the Last Gift study. Cheryl and Stephanie and I, we screen the inpatient lists at UCSD Hillcrest and even La Jolla, actually, but we screen our UCSD inpatient list. And that's sometimes often how we learn about patients. Occasionally, rarely, we might, sort of approach the patient ourselves as the study team with permission, usually of the primary provider or the, inpatient team. And I'll talk about sort of our recruitment efforts and sites and where, we have success and more where some are more successful than others. But I would say when we're looking at the inpatient sort of list, it's really to learn about people who might then follow up with their provider, where we can then, sort of see how they're doing later. We also have a database query for any patients with HIV who are referred to palliative care or hospice. So what I want to present now is sort of, some of the heart of my portion of this talk is, who are the people we're recruiting? And so what we did was we actually sort of tracked this over a year, starting like last September to the current August. And so we identified 28 study candidates through those methods that I just described. Of those 28, we were able to enroll 9. And so just to sort of characterize that group a little further, 6 of those 9 patients who had enrolled did so after discussing the study with their primary care provider. So 5 out of the 6. And 1, it was actually a conversation with their oncologist, Dr. Erin Reed, with whom we work very closely. She had the initial discussion with the patient. 2 out of those 9 were successfully recruited in the hospital, but 1 was already enrolled in a related research study in the CNTN, so kind of already knew about the study. And this was a group that was already engaged in research. And 1 of those 9 patients had actually self-referred via the website. We've, among this group, 2 have already undergone autopsy. So, let me talk a little bit about the characteristics of people who didn't enroll. Those were 19 patients. And I sort of excluded from the analysis 4 who had a primary provider who was a study investigator. I think in 3 out of the 4 cases, it was me. I'll come back to that point in a little bit. But so of all the non-Deiss providers who, had a potential patient, there were 15. And I just want to show you what happened with those. 3 of the participants were actually able to have that discussion with a provider. They declined enrollment, or declined any interest in the study. Our providers aren't necessarily, telling them to, about the study per se, like in order to enroll. They don't do the enrollment, but they might mention, would you be interested in something like this or in research in general? And sometimes the answer is a quick no. Of the other participants, so there were 12 where the providers weren't able to discuss, even the study conceptually with the patient. 2, because some of the reasons here were because of insufficient time during the visit. I think we can all relate to that. Provider might have felt the patient wasn't ready to discuss sort of end-of-life topics, or that it wasn't appropriate based on their current medical condition, i.e. someone receiving like curative chemotherapy rather than palliative. Sometimes there was no opportunity to discuss the study. Patient was hospitalized or missed their appointment, or someone's psychosocial situation might have precluded study enrollment, such as a severe substance use disorder, unstable housing, someone who's not reliably coming to appointments, etc. This all brings up what is kind of the dual role of the clinician researcher. I mentioned sort of my own involvement in this study, and sort of often seeing many of the same patients we're potentially recruiting, and sort of all the providers we've talked to for this study and have been amazing and very open in many cases to discussing the study. We're very grateful, enormously grateful for that, because it is a little bit of a burden on on a provider, because of this dual role, where a clinician might be, sort of considering research and advocating it, might have greater awareness of the benefits of research for individual patients and public health and scientific advancement. How do you reconcile that then with your clinical duty to the patient? There's some ethical implications and things that often need to be thought through. Are providers being asked to provide a research-related service because of their clinical expertise? Hopefully, no one feels that anyone is being taken advantage of in that realm. But these are always possibilities and questions. When a clinician brings up a research study, is that sort of providing an endorsement? And that's something that we have to carefully consider. And last, will a patient be able to distinguish that sort of study from their clinical care? I think people do an excellent job, but I just want to stress that it's not always easy to sort of navigate those dual roles. And so here, what I just want to show is that when sort of our 28 candidates, of our sort of 28 people that we identified, when the primary care provider was able to discuss the study, and again, just sort of mention it as we have this study, would you be interested in learning more? 6 of 12 individuals actually enrolled in the study, 50%. When it was a member of the study team, either approaching someone in the hospital or not, it's a much lower success rate, 3 out of 11. And as I mentioned, 2 of those were actually engaged in other research studies, so we're already open to it. And this actually counts the one self-referral as well. So really, having a provider sort of discuss with the study has or at least mention the study to patients has been sort of very effective for us. And, I hope that we'll show you some slides a little later that show that there are some benefits, you know, to participating in the study in terms of a patient's quality of life. So it is something that's, you know, I want to say not done in vain, and you know, and we're very grateful, and it's can be very important for somebody. 5, as I mentioned, weren't able to have the study discussed, you know, for any number of various reasons. And so where are our patients coming, or where are our participants coming from? 9 have been patients at the Owen Clinic, and that accounted for all the enrollments during this particular period of analysis. Of the non-enrollments, again, most have been, you know, Owen Care patients, but a few from San Isidro, 1 from the VA, and 2 others from, you know, 2 from other facilities. But that doesn't mean to say all the patients in the last gift study have been Owen. We've received some from Family Health Centers, Scripps, AHF, you know, and even other locations, but just not in this 1 year that I was looking at. So in terms of our sort of lessons learned and conclusions, again, primary care provider, a member of the care team, you know, often that's, you know, sort of provides the most successful approaches, sort of brings up that question of the dual role. But, you know, again, motivated patients, you know, it's often a very accessible way for them to learn about the study. I'd say that outpatient settings are better than inpatient just because of everything that's going on. And this isn't really that surprising. But even our Owen Clinic has, you know, sort of the unique collaboration with AVRC research, and, you know, people are familiar with research, and we see most of our enrollments that way. Also having me, you know, work in the Owen Clinic and learn about patients, you know, facilitates sort of word of mouth. And I just want to obviously add, and this has happened with me as well, but, you know, you might not have the time or the opportunity to discuss a study at this very sensitive moment of life. And so that's very well understood as well. Okay, one last section for me, and then I get to turn it over to Sara. There's a social and behavioral component of this study, again, that you know, people might not intuitively know about or realize if you're just thinking about it as an autopsy study. But the goal basically of this sort of piece is to better understand the experiences of loved ones or family members and participants in the last gift study. So this is achieved through interviews, questionnaires, etc. We like to collect information about their perspectives, and, you know, sort of their life histories, etc. And, you know, sort of get their opinion on cure research, talk about the protocol and how it can be improved, etc. The lead investigator for this piece is Karina Dubé. She is going to be presenting at High Rounds in several weeks. So more to come on this, but she's got a ton of research, and you know, Last Gift is only one part of it. These have been some of the papers that have been, you know, sort of have grown out of the behavioral component, you know, whether it's sort of just ethical considerations, and, you know, sort of the, you know, sort of qualitative studies on the perception of next of kin enrolling in a study like this. I love this quote, you know, "My death will not be in vain." This is a testimonial from one of our research autopsy participants, etc. So it's been a very prolific group. Karina is actually a bioethicist, and so it's been very critical to have a bioethicist on the team when we discuss sort of certain situations, dynamics. You know, it's critical to have her perspective. So one of the studies that she's currently submitting, actually, to JAMA, is a study where we looked at what was the effect of the Last Gift study on a person's quality of life. As background, we know that older people with HIV tend to report decreased quality of life as age advances, and this is also true of people without HIV in the last few months of life, sort of a decreasing quality of life. So our research question was, what is the relationship between quality of life and depressive symptoms and our study? does our study sort of have any effect on some of these measures? We looked at mental health via the Beck Depression Inventory, a sort of standardized instrument for quality of life and mental health questionnaires, and I'll show you some of the statistics here. So it's a very noisy slide, two of these, but what I want to show is that each of these dots represents a participant, right? And so this is their quality of life score sort of over the term of the study. I mentioned that some of the participants live, the median time on the study, if you recall, was 3 months. So some of the participants are living out very far. And so we also looked at, well, what was the effect of the study on their, depression or, mental health rather. So we had 35 participants who completed the baseline questionnaire. That was everybody. 24 completed a follow-up interview. Remember, some of these, deaths followed enrollment by only a few days. So, and in a few cases, someone wasn't able to provide, just, the behavioral questionnaire, let's say. But of the people who completed all completed the baseline questionnaire, but not everyone necessarily was able to complete the follow-up. And so of those, people either gave anywhere from 1 to 9 interviews. What we found in our baseline questionnaire was high levels of depression, almost 38%, and anxiety. This is something we know about people with HIV and mental health, and it's reflected in our study. So here's sort of the bottom line, is that despite this being someone's sort of end of life, and despite the fact that quality of life usually deteriorates in the last few months, in our study, these quality of life scores actually remain somewhat stable over time. And as did the depression scores down at the bottom. You'll see this trend line and it's going up, but really that's all driven by a few participants. So I think when we look at, when we adjust the model for that and sort of, look at, sort of where the score is, the line might be a little more flat, but it again shows that it's not falling, right? And again, and the same for depression, that people are not becoming increasingly more depressed at the end of life who are on our study. Now, does that mean the study is the causal sort of factor? Obviously not necessarily. We could be just seeing that our participants are different than others who are not participating in a research study, right? But I do think I think the bottom line, though, is it does suggest that there's something novel or interesting about the last gift study and our participants, sort of reflecting this this measure. Okay, so next I get to turn it over to Sara, who will blind us with science. There's always music in my talk, so Sara, I'll help you advance. But please go ahead.
Okay, well, thank you so much. So this particular presentation was not so much about the science. It was really, about telling you all of the effort that we have done with recruitment. But we thought that it would be nice to give a little bit of a taste of the type of data that the last gift can help generate. And then I can always come back and dedicate a full hour to present the scientific finding. But so basically, this is our last gift debut paper that was published a few years ago, where we characterize the HIV reservoir across multiple tissues in 6 people with HIV. It was published in the Journal of Clinical Investigation. And you can move forward. Mm-hmm. And here, one of the things we did is we measured the level of HIV DNA across tissues in these 6 people. And here you can see a few examples. For example, on the left, the high panel here, this was one participant, and he had a very high HIV DNA level in the pancreas. And it was interesting because he actually had pancreatic cancer. And then the second participant in the lower panel, had higher level of HIV DNA in the gut and in the lymph node, which was what is the more usual as expected. So, every participant has its own story. And then what we did, we also sequenced, the full envelope gene for all of the participants. We create phylogenetic trees, and we can see, how the virus in the different compartments relate to each other. Here you can see one tree for one participant where all of the different colors are different locations. And you can see how some of them are completely intermixed. And there isn't, Robert, if you can show, there is one particular, branch of the tree that show a clonally expanded population of virus that seems to be found across multiple tissues. And this, it's very interesting because it's an indirect evidence that there might be some cells that migrate from one place to the other carrying the same virus. Using those sequencing and some sophisticated statistical modeling, Antoine was able to look at viral migration across tissues. I think that what makes the last gift so special is that by following people so closely before the time of death, we are 100% sure that people are suppressed.
Excuse me, for hydrocortisone cream and some
Susanna, can you mute? Sorry. Because we run viral load often the day before autopsy or even the day of autopsy. And so when we measure at migration and we observe migration, we know it happens at the time of viral suppression. So you can see these are the very nice figure that Antoine generates where, we can see that there are some sequence that seem to migrate from blood to the lymph nodes or from, or even between from, lymph nodes to the liver, showing us that the viral reservoir, even in people that are suppressed, is not a static picture and that cells can move across compartments. This is a more recent paper, which I am really excited about. For those of you who have been interested in neuro-HIV and in studying the reservoir in the brain, you know that, and there are many of us here, and at the HNRC in particular, it has always been a little bit controversial, and there are always people saying that maybe the brain doesn't matter. So we had this collaboration with Dr. Yanka at the University of North Carolina, and he had developed a way to sort fresh microglia from cells, from brains, and then to cultivate them and grow the virus. And he has done it with a brain from primates, but so far was never successful to recover the viable microglia from brains from humans. And when he connected to the last gift and we started sending him some fresh brain, using the protocol that Robert just highlighted, where we drive, the tissue to LA and catch the first flight, so that most of the time the brain arrives in North Carolina, 8 to 12 hours later, and then they start immediately the processing. They were able to sort microglia from 4 participants, and you can see here in this figure that in all of them, they were able to find HIV DNA and also low level of cellular HIV RNA. So this was kind of the first evidence that there is indeed some HIV in microglia. And now they brought it further, and Robert, you can go to the next slide. What they did, they took this microglia and some T cells that they sorted from the brain, and they cultivated it with cells from people without HIV to see if the virus can grow. So here in the panel B, these are the days post, co-culture, and you can see how HIV starts to grow, and they were able to recover HIV from microglia from all 4 study participants. You can see it here in the panel C, but none of the cells. Now, of the T cells in the brain so they were unable to grow any HIV from T cells from the brain. And this is kind of the ultimate evidence that there is a replication competent reservoir in the brain. Now, obviously, reviewers are not convinced unless you prove that the virus that grows is really from microglia, not some sort of contamination from blood. And so if you look at the next slide, so to prove that the virus is really from the microglia, first we sequenced the virus in the microglia, and then we sequenced the virus in the brain, and then we sequenced the virus in the blood, spleen, and lymph node. And you can see how the virus in the microglia was very close to the virus in the brain, and it was not the same as the virus in the blood and spleen. And then in collaboration with Dr. Sara Joseph, who has developed an assay to show if a virus is T-cell tropic or macrophage tropic, she was able to prove that the virus recovered from the microglia was indeed macrophage tropic. So the reviewers were convinced, and the paper got published. And I like to say that this is probably the most compelling evidence that proves that there is indeed a replication competent reservoir in the brain. And this will never be possible without the last gift and the donation of fresh brain tissue and all of the infrastructure to send the brain so fast to our collaborator in North Carolina. So I wanted then here to show you just some screenshots of data. These are data generated by our collaborator at Gladstone Institute in Northern California, where they ran single-cell RNA-seq from cells from the spleen, and they were able to enrich for cells that carry HIV, so that if you look at all of the small dots, these are cells that are HIV infected. And you can imagine this is very important, because then if you analyze the cells that are HIV infected separately from the other cells, you might be able to come up with some characteristics and compare them across tissues. Again, to do this type of work, you need viable cells from tissue that are freshly collected. The same group also designed a CyTOF panel. So CyTOF is kind of a very sophisticated flow cytometry that has many, many more colors, like 40 colors. And here they ran their CyTOF panel on multiple tissues, and then they kind of pulled all the tissue together. And you can see here in the arrow, they found a population of CD4 T cells that looks very different and is only in the gut and in the male reproductive tract. And so they spent some time, looking at these CD4 T cells. You can go to the next slide. And they were able to prove that these CD4 T cells were enriched for some protein that are sensing viral replication. So that they somehow react to the virus in the gut and in the male genital tract. Again, this particular immunological assay can only be run on fresh tissues. So this, and you need enough tissues to be able to characterize the CD4 T cell. So again, this work will not be possible without the last gift. And there's a postdoc, a junior faculty there, Ashley George, who is writing the paper. And this has been presented at IAS as an oral presentation this summer. And then we have another collaboration with Dr. Colin Kiefer. He is at the University of Illinois, and he is like an expert in fancy imaging. I asked him to send us a few of his images, and this is intestine, where you can see he's able to image immune cells, and then in green you can see HIV actually in the cells. And then he also has another picture. This is a brain, again, from one of our last gift participant. And it's very hard to see. So we put some arrow here. You can really visualize HIV in the brain. And the beauty about the imaging is that you can really, it's preserving the structure. So if you look at the next slide, it's very provocative that it seems like that he finds much more HIV in the astrocyte than in the microglia. So, we put him in contact with Gauchon, and we were thinking that maybe the next step will be to sort astrocyte and to see if actually Gauchon can grow HIV from the astrocyte. You can see how the last gift not only provides tissue, but also create a network of collaboration so that people can kind of feed from each other's finding and advance the science together. So we have become really popular, and we have a lot of people that request tissues, and I'm really excited about it. So, okay, I just wanted to have enough time for Robert to wrap up. So I'm sorry if that was a little bit fast. I picked kind of the most exciting.
No, I agree. And I think you made a case. We're going to bring you back to do some more. So just going to wrap up to sort of show the next steps of this program, sort of the scientific, social, programmatic goals. Obviously, we're going to continue to characterize the immune environment and its connection with HIV persistence, epigenetics, and looking at actually the pharmacologic environment. We've sort of began to look at opiate use and how that might affect some of the milieu surrounding the virus. Again, our social component is very active, and we're looking at the ethics. I mentioned some of them about the dual role in research. We have the quality of life paper coming up. Programmatic sort of new goals. We're always trying to enhance the diversity of our study participants, and we just learned recently that a UCSF researcher reached out to Sara about maybe opening sort of a last gift study type program in Brazil, and that would be amazing, because then we'd get to look at different subtypes, clades, etc. Ultimately, the sort of future of this study is, right now we're observational. We're characterizing, sort of what's going on in tissues, et cetera. But, there's opportunities here for interventional studies, right? Having a participant interrupt treatment towards the end of life, and, what will happen with the reservoir with certain, cure strategies, et cetera. So, that's sort of the future of the study, and that's obviously very exciting. And then sort of our last new development that we want to get the word out about is that we recently received a supplement, a one-year award to enroll people without HIV into the last gift study as a control group. And the idea there is that's going to help us sort of maximize all the data that we already have collected, sort of in our in the research that we have to date. We'll be able to collaborate with other groups who do rapid autopsies. There is an ALS program, for instance, and other rapid autopsy groups around, for people without HIV. This opens up some new doors for collaboration. I also think that most importantly, this is an opportunity for next of kin, partners, or friends to participate in HIV-related research as well. In the unfortunate circumstance that someone knows or is close to someone, who is, passing away or approaching the end of life, this might be an opportunity for those individuals, and especially if they're part of of the community, we might have a potential sort of easier access or more buy-in from this group of individuals. It also sort of provides a similar study population, age-wise, and other factors from our participants potentially. And we continue to do community outreach and recruitment. So I'm sorry I talked more than who knew we'd that I'd talk a lot. But it's a huge team, and not just of the study itself, but also the collaborators, CNTN, our parent study, as I mentioned, all the people that go into a study like this, and obviously our most important acknowledgement to the Last Gift participants and their next of kin as well, who participate in this research, provide a tremendous gift for us again. And then we have our community advisors. They might even be on the Zoom today, Andy Cates and Jeff Taylor for the study. So thank you so much for your attention, and thank you, Sara, for that, amazing, sort of rapid tour of our findings. Happy to talk further if there's any questions from people or anything discussion.
Great. Thanks, Robert and Sara, for a wonderful presentation. I guess we can open it up, but I do have one quick question. So if someone's interested in referring to the Last Gift, can you tell us how we or who we should call and what number to call?
Sure. Yeah, I gave the website link, and whoops, that's the next slide. Yeah, I can. I think, Sara, the number we're giving out right now is the HNRP number, because they do enroll in the parent study as well. So that's 619-543-5000. We can put that in the chat. That's the number that will get sort of that someone would see if they visit the website. I'm happy to sort of be the initial point of contact for anybody if they have questions from a provider standpoint. Just reach out to me, and I think my contact is all over the place. So but RG Deiss, I'll put it in my own chat box. I think that's the easiest way to do it.
All right. Does anyone have any questions? You can unmute, type in the chat.
Yeah, no, as someone, Dan, who's also had these conversations with patients, I might, just sort of pick on you. And again, we're grateful. I think you've provided a few referrals. And is there anything that you have found when you talk to patients about sort of this study and what that conversation is like?
Yeah, no, I think in my experience, it's most of the time it's been more the patients that have kind of approached me initially, and they were thinking about kind of end of life things, and they were like, for instance, there's one patient that was trying to arrange for funeral services for his funeral at some point, and had questions about if I get involved in a study like this, will that cover for funerals, services, cremation, those kinds of things, and was looking for other avenues. And so it was kind of an easy kind of to then have more of a discussion about, what kind of studies were available. And so, but, I think in circumstances where that doesn't come up, I think I do try to bring it up when someone is seems a bit more terminal, and I think the conversations have gone fairly well for the most part. Obviously, I think it requires being sensitive to their situation and getting if you know the patient well enough, I think you can have those conversations. So I do think that having the primary care provider involved is key.
Yeah, no, and that's what we found. And sort of as again I mentioned that it's sort of a mini analysis that we did, and eventually we'll write that up. But it's that was, as I mentioned, the overwhelmingly most successful approach. And again, expressing my gratitude and acknowledgement for our Owen team and community providers who have these conversations with patients and yield these.
David, did you have a comment?
Yeah, I just wanted to say that when in doubt, refer is my suggestion. So certainly the people may be eligible for the CNTN study, which would then directly flow into the Last Gift. People can be co-enrolled in those two studies. So if you have any patient or potential participant that you're thinking about, please, send them along, and we will, do our best to get them enrolled in the parent study, which doesn't require that people be as imminent as last gift. And then if they become imminent, we would, kind of collaborate, and people can be co-enrolled in both studies. So I just wanted to make that clear to folks that when in doubt, please refer, and we'll do our best to get those folks enrolled.
Great, thanks.
Yeah. And I think it is important that as Robert said, study participants really benefit a lot from being enrolled in the last gift in terms of, network and support. And we have a wonderful outreach team. Everyone loves them. Usually participants get close, to Cheryl, Stephanie. They're amazing. Susanna now is gone, but she was like the heart and soul of the last gift for so long time. And we have so much experience also supporting people with, as you said, right, putting together their last will. We do pay for cremation, not for the funeral, but we do pay for cremation. So I think that we there are some benefits of being enrolled that are beyond just donating the body.
Great. Scott?
Just from a scientific standpoint, I thought I'd bring up that, we work with a lot of neuroscientists, and people know how to grow astrocytes. So I attended 2 meetings this week with groups from one from Sanford Burnham and one from Rush, growing astrocytes. So, for some of these things, you don't need to reinvent the wheel. If you, reach out, we can help connect you to scientists who do that all the time. So,
yeah, thank you, Scott. You definitely, yeah. We're just used to work with Gauchon because now he is our brain to go person. But, I agree. We have wonderful brain people here.
Yeah, you got a whole group of brain people here.
I know, we don't have to go to North Carolina.
Great. Well, it looks like it's a little bit over time, so I wanted to end today's session and certainly thank both Doctors Deiss and Gianella Weibel for their excellent talks. And we will hopefully see you next week at our rounds. So thank you. Have a great weekend.
Thank you all.